Hello everyone, I'm Zhao Zhiming, and I'm from Professor Kuan-Fu's lab. Today I'm going to talk about my project this year, and the title is Analysis of Variability-Based Prediction Models. In this model, we're building the species and the HRV models. There are studies that confirm that the level of HRV is related to death from species. And in the study patterns, there are adult patients who visit the ED with an infection. There are total 38 patients which enrolled with total recording time of as much as minutes and resulting 4,314 analytical units. And in this analytical units, 12.5% met the species 3, which means their total score is bigger than 2 after 120 minutes. We want to see the different interaction with HRV and different physical markers that relate to biological waves. And these markers is from here. And in our lab period studies, this one FFDP markers show the best performance in predicting species 3, and in SHARPie additive explanation, lower values of FFDP have negative association with species 3 onset after 120 minutes. So my research is mainly the extension of our laboratory study. I want to see if HRV has a different performance under different time and different markers. It's because empirical research in a laboratory only use like 120 data to make the model. So this time I want to see if there has any change between leg 30 to leg 120. So by using the purest data in laboratory, I made 3D graphs of each variable and their shape to see if there's any trend between them. So I totally got 160 graph here, and I want to pick the graph that has like a trending here, not like this. So totally I pick these 3 markers that their picture seems have a trending here. And this data set looks like there are totally 3,000 and 2,036 objects and 20 clones. And this code look like. And here's a GIF look like. And this one is AFDP. And make them separate look is a little bit difficult to look, so I put them together to make a one GIF. And this like this. This one is FDP, this is YFL, and this one is the parallel slope. And as you can see, these they do change a little bit with over time, but their basic trend and the shape are roughly the same. And to guess what caused this change, this is a data set look like that the patients have been tested their biomarkers, And in this paper, they have said that these kind of biomarkers have it has a relationship with inflammation, and I want to put them into the FDP model because FDP model has better performance in predicting species 3. And this is how the code look like. And here's the result. Except IL-6, the red dots are concentrated in a certain area and the position does not change too much over time. The IL-6 is based on a more even coffee-colored dots distributed throughout the entire GIF. So based on this picture, I think the biomarker that most likely to affect the entire model is IL-6. And their study proved that species 3 is the case of increased biomarker of IL-6. Also, another study showed that the increase of IL-6 will cause a drop of HRV. A low HRV can make inflammation even worse. So it's the timeline. So I think IL-6 is like